Thank you, everyone, for joining us online. We definitely appreciate it. Really delighted today to have two experts in artificial intelligence joining us to talk about how they're applying this technology, this emerging or rapidly emerging field, to the problem of drug development, drug discovery. And so wide-ranging topic, and we're just going to touch the surface on it today. But leading us off today is our colleague from Texas A&M up at College Station, Dr. Yang Shen. So Dr. Shen joined TAMU 2015, so he's been there a few years, into the Department of Electrical and Computer Engineering, which doesn't sound like he should be doing drug discovery because he's, over there and not in a biology department. But these technologies really cut across everything. So Dr. Shen, before coming to Texas and joining us, got his BE, science technology from China, then went to Boston University, and then postdoctoral training at MIT, and spent a little bit of time Toyota Tech Institute in Chicago, and then said, Texas is calling, right? And here he is. We're delighted to have him. We hope to have him more closely integrated with the biology community in Houston, because College Station isn't that far away. So Dr. Shen, thanks very much, and I turn it over to you.
Thank you very much, Stan, for the generous introduction. And thank you, Susan and Stan, for the invitation. Can you hear me fine? Is voice and...
Perfect.
Okay, thank you very much. It's a great honor and pleasure to be here today. I mean, being in College Station and being in Texas and having access to the great experts and talents here, coordinated in a consortium like this and working together for innovative drug discovery and development. That's just amazing. So I really look forward to hearing your feedbacks and getting more relevant and impactful research toward this direction. So today I'm going to talk about two topics: explainable deep models for binding affinity prediction and deep generative models for molecular design. So our view from a relatively naive engineering perspective is that just like man-made systems, the biological systems are fundamentally hierarchical. We have bioparts, biodevices, and biosystems all the way. So the way I look at my research is that I'm going to focusing on developing algorithms for both reverse engineer the biological systems and forward engineering such systems. So we do a lot at the molecular level, for instance, modeling and designing molecular interactions. So the goal is to not only probing the inner workings of biosystems, but also engineering utilities for biosystems. So speaking of algorithms, one element in the room is artificial intelligence. And then speaking of molecular discovery, one of the most exciting molecules we can discover is drugs. So putting these two together, you can imagine what kind of chemical reactions can occur. So indeed, a lot of expectations and hypes are coming in from drug discovery, for instance. So the impact of AI is making rippling effects now, and no doubt it's reaching drug discovery. And at the same time, right, as an algorithm developer, I keep thinking about this comic, right? So I don't know whether you watched this meme, Here to Help. So as an algorithm developer, I try to keep humble, and I believe that this problem is challenging enough that there's no magic that somehow a technique just occurred somewhere else and then get directly translated into a new field and can solve that problem. So that's my view. And indeed, right, so Aaron is going to talk more about it. But some of the roadblocks you see, despite the expectations, right? So include limited acceptance, right? As much as we would like to make an impact, still there are limited acceptance. And also at the same time, from us side, computational scientists, I have to acknowledge that a lot of the active research is probably still in the low-hanging fruit part. So we're probably working on a lot of problems, including my today's talk. They are not going to, they are not really the bottleneck of the 10-year drug discovery process. It's not necessarily the bottleneck of the low discovery or approval rate. So how do we go for focusing on the right things? So that's part of the reason I'm coming here today, and hoping to get more involvement and collaboration or interactions with this consortium, because we definitely want to focus on the right things and make more impactful discovery. And I believe that the innovation comes from across disciplinary interactions. So, so our perspective is health informatics targeted AI, but also the health informatics inspired AI. So, so I'm going to talk about two topics. One is the forward problems. Okay, the forward problem is that you start with the input or cause, in this case, a molecular identity, and you are trying to predict a output or effect, in this case, a molecular property. So this system we are having here could be experimental approaches, could be computational approaches, could be AI technology. On the same side, we also have to think about another kind of problem that is probably more relevant for drug discovery. That is the inverse problem, that we start with a desired output or effect, and we're trying to find the molecules or causes that is optimized toward the desired property. For instance, for drug discovery, you're targeting some purposes. And then that is probably more relevant. So that's the second part of my talk. And the first one is a forward problem. Is we are talking about a discriminant model. The question we're trying to answer through this model is, how do we quickly predict protein-ligand interactions at the genome scale with interpretability? So that part is about 2/3 of my talk. So we're going to first focus on the prediction, but then talk about interpretability a little bit more. In the second part of the talk, I'm going to talk about the generative model for an inverse problem. So in this case, I think I'm asking an overly general question, and my current approach is absolutely very, very humble. And but the question we are asking is how to directly design compound combinations. I'm avoiding drug combinations because it has to be FDA approved to be drugs, to specifically target a disease module. So, and to answer these two fundamental questions, our current approach is the integration of data and knowledge through deep learning. So definitely we need to hear more from the knowledge side and from experts, audience like you. So the first problem, discriminant model for a forward problem. So here we're looking at this problem, right? The drug discovery is not targeting a protein, right? It's targeting human, right? Where human is a biological system, as we said at the beginning. So there had already been a paradigm shift from one disease, one target, one drug to a systems pharmacology. You actually have to hit desired multiple targets, and then you have to miss those undesired multiple targets to avoid toxicity and side effects, right? This is for single drug even, right? So if you think about drug discovery, we are focusing on protein small molecule interactions, looking at the currently FDA approved drugs, human drugs. So if you think about the space we are talking about, right? So for the compound side, we have about 10 to 60. That's the estimated size of the chemical space, okay? From the human side, if we talk about the proteome, or specifically the proteoforms, because there could be different isoforms, there could be different mutants, the expression level, and so on. At least we're talking about way more than 10 to 6 different proteoforms, right? So we have all these combinations of proteins and ligands. when we have to consider a drug effect in the biological system. At the same time, for each such combination, compound protein combination, we have to think about a lot of properties that can have to be assayed. For instance, the IC50s, EC50s, KIKDs, and other pharmacokinetics and dynamics, and a lot of the combinations of protein, drug, and assays that have to be done. So it's daunting, if not impossible, to do every combination experimentally. So that's a perspective. We think that probably there's a need for computational prediction of compound protein interactions, not to replace, but to facilitate the discovery process. So looking at the prediction of protein drug interactions. So what are the current methods and what are their pros and cons? So one kind of the classical way is, starting with the protein structure, you dock molecules into it. So that's where my training started with, and it can predict the activity level. For instance, you can use energy model, free energy models to model the binding affinity. It's very interpretable. You can decompose the atoms and the residues that are in charge of these interactions, right? But the disadvantage is we are talking about a free energy minimization, which is very challenging and slow. And not to mention that many protein structures are not available. If we look at the gap between the proteins that have been sequenced over years and those that are structure determined. I know AlphaFold 2 is probably bringing a lot of hopes, but still, even if you have the structures, you have to do the docking. That is very non-trivial, right? At the same time, the data-driven methods are coming, and their pros and cons are opposite to the pros and cons of the structure-based, okay? So they only use a sequence. So they avoid the use of the structures so that it's more applicable, right? For more generally applicable. So, but when we started back in 2017, I think most of the methods only just try to classify into two modes, whether they are interacting or not interacting. So it's a binary classification. And none of the methods was interpretable. They give you a score of 0 point something as a probability. They don't tell you why. But on the plus side, this machine learning is really, really fast, way faster than protein docking. And we have a lot of data now, actually, compared to the structure data. We have a lot of sequence data that have the binding affinity labels already. So that's pros and cons. That is totally opposite. So when we look at this problem, we were asking, can we get the best of both worlds, right? So can we get the realistic setting to predict the actual binding affinity, okay? Not just binary classification. And then can we achieve the broad applicability by just using protein sequences, but we keep a structural context? I will talk about it. And at the same time, we make it a high throughput, potentially a machine learning model. At the same time, this model better not be a black box, so it needs interpretability, right? So that's what we are aiming to fill. So how do we do it? The way is integration of knowledge and data under deep learning. So one component is to have informative yet compact molecular embedding. So the other one is we can use a lot of unlabeled data besides labeled data. So there are a lot of protein sequences with binding affinity, but there are a lot of protein sequences, way more, without such labeled affinity either. They can be utilized. And then we talked about a novel machine learning framework combining recurrent and convolutional neural networks for semi-supervised deep learning. And we embed attention mechanisms for interpretability, and I will explain in more details. So here we have the deep learning protocol called the deep affinity. And we start with the data representation. So we have the protein sequences or smiles or graphs of the compounds. And traditionally, we have a lot of compounds or proteins without the paired affinity, right? So those unlabeled data can be utilized. At the same time, we have a little less, but still around 500K labeled pairs. So, and then we cleaned this data set from that binding DB, and then we completely withheld 4 protein classes from the training. You may wonder, why do you exclude all these important drug targets, ion channel, HPCR, and kinase, and so on? We'll explain that later on. And then we randomly split the rest into training and test. So we test our models on the test set as well as the generalization sets. Okay. The compounds are usually represented in 1D fingerprints. And here we are using the smiles and graphs because nowadays, due to natural language processing and recently graph modeling, these inputs are getting more and more attention. For proteins, we are looking at one-dimensional sequences, and then we are using the sequence predicted properties. Then we basically combine these 2 dimensions and make words. So for instance, you have CEKS, which is a word for a protein secondary structure. So it means a coil exposed, basic, and small. So in this way, we have a structure property strings, which is shorter, easier for modeling. At the same time, it has a structure context from the sequence predictions. We use a sequence-to-sequence autoencoder used in natural language translation. And then in this way, you are basically embedding these sequences into a latent space and then translate it back into the original sentence. So that's why it's called autoencoder. So in this way, you can utilize the massive unlabeled data and then train this embedding. Once this embedding is initialized, you can utilize the supervised or labeled data. In this case, you have the protein compound pairs with affinity. So in this case, you can put attention mechanisms, which are basically weights on pairs of compound atoms and protein residues. So in this way, you can predict affinity, but also try to explain why the prediction is in such a way. So the first question we ask for this method is deep learning helping? So we can see that we have different set of ligand protein pairs, and then we are asking, can you compare the random forest, which is a very good shallow model, and your deep affinity? And here I'm showing the root mean squared error. So the lower the better. And in the parentheses, I show the standard, the Pearson's correlation, the higher the better, okay? So we can see that for various data sets, we're having better performances compared to a shallow model. Then the next question is, well, remember at the beginning, we say that we excluded a lot of protein ligand pairs involving nuclear estrogen receptors, ion channels, GPCRs, and so on. The reason is that people believe that deep learning heavily relies on big data. That is true. And then they get worried when they have a new protein target that has very little labeled data. Then they probably think that doesn't make sense to use deep learning, but I'm gonna show you that that's not the case, okay? So we're looking at these, for instance, nuclear estrogen receptors, and then we're looking at the dashed line. The red dashed line is the original, the original deep learning method, right? So it has a very high error, right? And then the yellow dashed line is the original random forest. Both are high. So the solid line is basically saying that, let me just use a small set of data just for nuclear estrogen receptors and make a random forest specific for nuclear estrogen receptors. As such data increase from left to right, you see that the error decrease and decrease, right? So that sounds very ideal. At the same time, we ask the following: what if you do the different way? What if you start with a meta model, meta deep learning model that was trained on data not involving nuclear estrogen receptors? So you started from basically here. Then you start to add more and more data specific to nuclear estrogen receptors and transfer your model, transfer your meta model. Then you can see that with the same amount of data, we're actually doing way better compared to a estrogen receptor specific random forest. So this shows the power of deep learning when even if you have a new target class. So then we also look at the drug selectivity and so on. But something I want to emphasize is that what about attention? What about the interpretability? So we sort of showed the attention on the protein side only. So this is a compound and this is a protein in cartoon. And the colors actually shows the attention strengths. You can sort of see that attentions are really more around the binding site. That was very good. So this was actually very good results to us back then. And that's the first interpretable model for binding affinity prediction. But then we ask more. Is the interpretability really enough, right? So we have to treat this problem really seriously. So in other words, you first have ask, how do you define the model interpretability in this context? Then you can ask, how do you assess such model interpretability? So to define model interpretability, you actually have to conform to the scientific knowledge, the human ways of understanding this phenomena, as well as the need in drug discovery. So for all those purposes, we think that a good way to define the model interpretability for affinity prediction is ability to explain predicted affinities through the intermolecular atomic context. By the way, they have ground truths already existed in crystal structures, right? So given those ground truths available, we can easily assess them, right? For instance, we can treat them as binary classification for the moment, and then we can assess the accuracy of the binary contact prediction, right? So using a ROC curve or a precision recall curve. Because the contacts are so only representing a tiny minority among all possible interactions, so it's imbalanced. So the area under the precision recall curve is a better performance metric. So we had to curate a data set involving 5,000 protein compound pairs. And then we have the training set randomly split with the test set. Then we have this new compound, new protein, and both new sets for generalizability test. And then we assessed our deep affinity model. Back then, there's only another interpretable method, Gauss-Settl, which was only for the binary prediction of protein ligand interactions, but we converted into the regression version for affinity prediction. As you can see that although the affinity prediction accuracy is pretty good for different models, but if you look at the contact prediction accuracy, which is the interpretability of the affinity prediction, they are merely more or less the same, just above random. So this results is a critical test to show that if you just ask a deep learning model to learn from the labeled data and additionally ask them to know where to pay attention, that sounds too much to ask, right? So then what do we do then? So beyond, realizing that attention alone is inadequate, so we have to define, we have to access, but we also have to enhance the model interpretability. So what do? The first thing is that you have to teach your machine learning models with examples where to pay attention, right? So this can be done easily through a loss function. At the same time, you have to teach models with knowledge where to pay attention. These can be regarded as regularization. So for instance, we don't have the protein structures, but nowadays there's very mature methods to predict the structure context from a protein sequence, right? So we can predict what is the surface and even the contact map, which residues are close to each other. So you can use the sparsity of the contacts, you can use the sequential proximity, and you can use the spatial proximity to regularize such attentions. And lastly, you can introduce explainability directly into the architecture, which I'm gonna skip for the moment. But basically, you can use the physics-motivated atomic relations explicitly written into the neural network architecture. And then you can increasingly focusing your attention from the predicted surface, predictive binding site, and the pairs, and so on. So with these three contributions, we have deep affinity plus and deep relations. And first thing we assess is how is the interpretability now coming? So the light blue is deep affinity plus and green is deep relations. And the dashed line you saw before, 0.004 was a random performance. Now we have 30 or 10 times more compared to before. So this is a great improvement of interpretability without sacrificing accuracy. And if you look at Gauss-Settle, for instance, the predicted interactions shown in dashed line are all wrong and quite scattered. Whereas in our case, you see more correct interactions. First of all, this context, the correct contexts are in red, and they are very focused, and the few false positives in this case are probably just pairing with the wrong partners, which can be just around the supposed partners. So you may ask, well, interpretability now you have, what is it to help? It can do a lot of things. It can do the sequence based or structure-free binding affinity prediction. So binding site prediction with high accuracy. And it can help your structure-based protein ligand docking. So if without any knowledge to optimize the autodock VINA, you probably have this quality of docking results in angstroms. The lower the better. But when you use our contact predicted from the deep affinity, remember the accuracy is, AURC is only 70%. You may think if you think about AUPRC, or in the top 10 that we only have 5. But even with the false positives altogether, we can improve the docking performance much more. We can also assist the structure activity research by improving the ligand scoring. So this is a D3R challenge involving the prediction of subtle changes of different ligands and their corresponding affinity changes. So we compared our methods post hoc to about 18 structure-based methods. We don't use any structures actually. And we can have, I think we outperform 2/3 for JAK2 and outperform every structure-based method in the type 2. But we didn't just ignore structures. I think we already incorporate the structure context, but just predicted from the protein sequence. And then we can also use this analysis for lead optimization as well.
Yang, five minutes left.
Yeah, so lastly, I will summarize with the inverse problem. The inverse problem we are focusing on is, drug combination. So you may ask, why do I need a different model called generative model for a inverse problem? Why don't I just use the discrete model going forward, and then I apply that model on every compound in the library and figure out which one is the best? Well, the problem is that you're finding a needle in the haystack. You may think that deep learning gives you a magnet, but it's very slow to finding that needle in the haystack. You may as well as just generate the needle directly based on the desired properties. So I think there was a paper already published in 2019. Of course, we can argue that it definitely is not the case that AI discovered a drug. A lot of things that remains to be done. At the same time, what we are focusing on is drug combinations, because this is important to lowering the drug resistance. It's a well-established systems pharmacology approach already. And you need generative models, as I said before, to generate new drugs effectively and following its supposed distribution instead of biased by a library. And what are the challenges? Well, designing a single drug is already challenging. If you define the combinatorials, it's more challenging. But conceptually, there's a gap because we have to target a disease module with low toxicity and potentially high resistance barrier. So how do we do that? So our answer is to use reinforcement learning for the generative model, and then use network biology principle for the design principle. So the way we do it is we start with embedding a disease, just represented in a way that a computer can recognize as a vector. And then given the disease, then we can generate drug combinations using reinforcement learning. And then all the drug candidates will be evaluated through the deep affinity through the process. And then the network-based reward will guide the drug combination generation. So, for disease embedding, what we are doing is that we have gene-gene interactions, we have disease interactions, and we have gene-disease associations. So we can use a hierarchical embedding to have disease embedding that consider all these network relationships. So secondly, for reinforcement learning, we designed specific rewards that will reward the chemical validity, that will also reward them to resemble the actual drugs. And at the same time, they are rewarded if they follow a network principle that is discovered by the Bravasi group back in 2019. They're saying that if two drugs are separately, right, targeting the disease module, which is gene-gene interactome responsible for the disease, and if they don't have any overlapping, that's called complementary exposure, and then they can jointly cover the disease module, then that would be ideal. That's the least side effect and the most clinical efficacy. So we found that based on our method, first of all, the disease embedding we have is much more accurate than others. And more importantly, our design pairs follow the network principle, mimicking the drugs. And when we evaluate our designed drugs over 4 diseases, right? One is a network score and one is predicted toxicity. And there's no baseline method we can compare. So we have to compare to random combinations or approved drug combinations. But basically you can see that the red ones are ours. They are more following the network principle to the right. And at the same time, they are less toxic. They are more to the left in the toxicity. And at the end, the designed pairs actually reproduce approved polypharmacology strategies. We can figure out who are the targets. So with this, I will end my talk. So as a summary, I talked about both forward problem and an inverse problem. So in the first case, I think what you can do here is that we not only give a forward prediction, we also give a molecular mechanistic hypothesis that can be directly experimentally tested. At the same time, it's the same at the systems level. You notice that our network principle is translated into a reward. So if you have a different hypothesis at the systems level, we can translate it into a different reward and design molecules that can directly, the optimal molecules that can directly test your hypothesis experimentally. With this, I'd like to thank my students who are making all this work possible, as well as my collaborators, especially my experimental collaborators who put a lot of trust into computational scientists and inspired a lot of research here. And thank for the funding agencies, and thank you a lot for your attention.
Thank you, Yang, for a great talk and to getting us started in this area. And there's a lot to cover. Now we're going to switch gears and we're going to go across the Atlantic. And we want to thank Dr. Morris, Aaron, for joining us. It's late where he's at. He's either at Oxford or Cambridge or in London. I can't remember, but he'll remind me. But you know, Aaron is CEO and co-founder of Postera. He is one of these really exciting up and coming AI-based firms that bio tech companies and pharma are counting on to really help them move into the future. So Aaron, mathematician by training, applied mathematician, completed his studies in Oxford University around 2014. And it was there, you know, probably in a dorm room or over coffee or beer and at the university when we could still do that he met his co-founder. So I don't know how the future is going to be for meeting co-founders, but at least Aaron was able to meet his co-founder at Oxford, spent 5 years after his studies working in finance as a data scientist and trader. And then last year, or almost 2 years ago now, he and with Alpha Lee, they founded Postera, and they've had really great success in the last 18 months with this company. And he's just going to touch on some of what they do, probably not all, but please reach out to him, maybe not right after this talk since he's going to go to bed, but maybe tomorrow. So Aaron, I turn it over to you.
Thank you very much, Stan, and thank you, Suzanne, for the arrangement and the invite here. Also, thank you to Dr. Shen for giving a really insightful overview at some of the latest advances. So just for the next 25-30 minutes, my talk is going to try and focus on some of the actual applications of how we have been taking the latest advances in machine learning for drug discovery and applying them to some of the actual practical problems that you will face when trying to make a drug to cure patients. So let's hope the technology works for us. I am going to share my screen here, and then we'll kick off. So Stan, just give me a shout if you can hopefully see my presentation.
Perfect. Excellent. Thanks.
All right. Fantastic. So in a nutshell, Postera focuses on applying machine learning to a very specific context within drug discovery. That context is referred to as medicinal chemistry. There is much that can be said for the uses of artificial intelligence across the entire life spectrum of developing a drug and ultimately putting it on the shelves or in the hands of clinicians. However, Postera focuses on what we call preclinical drug discovery, and you can effectively characterize this as everything before going into human testing. So preclinical before you're going into the clinic. That entire life cycle of preclinical drug discovery is where my company focuses on applying the latest in machine learning. So at a very high level, what does that preclinical stage look like? Well, it's typically characterized by a three-step process. You've heard a lot about one or two of those processes from Dr. Shen, and it really starts out by designing compounds that can be effective against a given target. It can be a foreign target in the body, such as a bacterial infection, or it can be a human-based target, a given protein in the body that is hypothesized to be relevant for a given disease. So you start out by designing molecules. And I would say back in kind of 2012, 2013, when the first emerging startups began in this space, that was their real emphasis. It was, how can we move away from chemists drawing molecules on a whiteboard? How can we have machine learning and algorithms begin to generate novel chemical structures? And so that was a lot of the hypothesis. What molecules should we make? But after you've decided what molecules that you think will be effective, you've then got to figure out how are you actually going to make them. This is a process typically in the small molecule space referred to as chemical synthesis. It's where Postera spends a lot of our time, and it's so an area I'm going to talk in a bit more detail about it. It nicely kind of dovetails actually with Dr. Shen's presentation. Finally, when you have made your compound, you then test that compound in an assay to validate that it does what you expect it to do. Now, Postera as an organization has developed artificial intelligence algorithms that cover all three of these particular stages. The founders of the company published these algorithms in collaboration with some of the world's largest pharma companies like Pfizer and AstraZeneca. If you go on our website, they are available on there for you to read. And we think it is incredibly important, as Dr. Shen commented, that algorithms do have a level of interpretability, explainability that gives their end users a degree of confidence into why I should trust the actual predictions of an algorithm. We spent a lot of time invested in that area. Again, everybody loves benchmarks and state of the art. So here's a couple of charts showing that across the design, make, test cycle, the actual technology that Postera has published is generally regarded as state of the art. For those aware with some of the kind of traditional AI approaches, we obviously benchmark against good baseline models, both from the kind of graph convolutional neural nets, support vector machine, naive Bayes, as well as random forests, actually not in here, but it's also a very good shallow baseline. I'm going to talk about this second part of how you do chemical synthesis and make molecules. So I'll come back to that in a second. But again, the final thing I'll just notice, if you look at the test stage, you'll notice that you have what is actually or appears to be error bars around the actual predictions. One of the kind of critical breakthroughs that Postera offers is a principled approach to measuring model uncertainty. Experiments are very expensive, and so you want to have a really good grasp on how confident is my model in making this prediction. And you want to know that for two reasons. The first reason is I should know when to ignore my model because it's telling me I don't have sufficient data or I don't have a sufficient representation of chemistry to make a good prediction, so please ignore me. But secondly, and even more importantly, having a prior that machine learning is a valuable tool, uncertainty effectively tells you what is the data that your model is screaming for. What is the actual uncertainty that your model is saying, I would like to receive some data in this particular part of the chemical space in order to increase the certainty of my predictions. This is a process known as active learning, and it's something that we deploy in Postera. And what you can effectively see is that the error bars within Postera's models are much more reasonable in terms of when you should ultimately believe the model. Practically, how do we work as a company? Well, we work with different organizations to help them develop drugs. We take our machine learning technology, we partner with very large organizations like Pfizer, and we partner with brand new organizations, new biotechs with only 5 or 6 people. In the case of Neurolucent, we're collaborating with them to develop a small molecule for Alzheimer's disease. And so the idea is that we can take this technology and actually partner with organizations to help you advance your drug discovery using the machine learning innovations that Postera has. But actually for the next 5 minutes, maybe 10 minutes, I just want to focus on this bottom initiative, which is called COVID Moonshot. COVID Moonshot came about around 9 months ago. Postera launched this initiative and now leads this initiative. And the aim of COVID Moonshot is to effectively crowdsource a COVID antiviral. That's quite ambitious, and particularly in an industry which prizes IP, intellectual property, patents, security, confidentiality. Why would anybody do anything open source in the field of drug discovery? But what I want to do for the next 5 minutes is talk to you about innovations not only on the machine learning side, but innovations in the way that scientists are collaborating to find new cures for patients. The genesis story. COVID Moonshot began when Postera saw a tweet that was put out by the University of Oxford, a team within the University of Oxford called Diamond Light Source. And what Diamond Light Source had done was they ran a fragment screen. For those who are not aware, a fragment screen, you can kind of think of it as mini jigsaw pieces of drugs. So not fully fledged drugs, but like mini pieces of drugs. And they basically fired experimentally these fragments against the main protease of SARS-CoV-2. Protease is an important enzyme that allows the virus to actually replicate. And so what they found was about 70 or 80 different fragments that bound very nicely within the main protease, within one of the key pockets. And what they realized was this was a really, really great starting point for being able to take this fragment screen, turn it into an actual drug candidate. So we basically saw this data, we put it online, we made it available, and we said, hey, would anybody like to contribute their ideas for how to take this fragment screen and begin to turn it into an actual drug, an actual antiviral? I think, to be honest, 50, maybe 100 ideas from scientists. I mean, our company was about 6 months old at the time, so nobody knew who we were. What was quite amazing is 9 months later, we actually received well over 15,000 submissions from 400 scientists around the world. And more importantly than that, actually 30 different global organizations have supported the effort of COVID Moonshot in taking these ideas from just designs on a website to actual drug candidates that we are now synthesizing in labs and experimentally testing. And I think this just speaks to the advancement in an interconnected world where people can surely contribute their to. This is just some of the organizations that support COVID Moonshot to date. I think the really important point, again, just to mention, is that every single one of these organizations is working pro bono. All it allows us to move very quickly, but it also ensures that the developing world, who generally can't afford expensive experiments, expensive in the West, too can get access to the antivirals that we develop. And so this is a 6-month snapshot from the time that we started Moonshots. This is 6 months in, so there's been some progress since this, but within 6 months, we were able to go from a fragment screen to actually selective potent lead compounds well into the nanomolar range within just those 6 months. These were experimentally tested. In fact, 2 different biochemical assays were run in order to validate these results. And not only that, but the actual compounds that we developed, we then moved into live viral assays, and we were able to show that the antiviral activity of our compounds who were able to kill the virus in live human cells was comparable with remdesivir. Since then, we've improved on these results, and we're now actually currently testing our compounds in animals in vivo. And the ambitious aim of COVID Moonshot is to begin to nominate a candidate for IND-enabling studies, which is the kind of process leading into human trials, by the end of March. This has been a remarkable effort by well over 30 organizations, and as far as we are aware, this would be the first time in history that a drug has been crowdsourced from scratch. But I want to focus on Posteera's particular contribution to COVID Moonshot, and this is the problem of how can you address the problem of chemical synthesis? How can you figure out which compounds you should be making? And then more importantly, how do you actually make them? And you can think about this as almost designing a recipe for molecules. You start out with very simple chemistry, building blocks. How do you combine them to create drug compounds that are obviously more sophisticated chemistry? So Posteera's approach is that we actually use a set of algorithms that are inspired by natural language processing. There are many ways that you can represent chemistry in molecules. One of them is you can represent chemistry as a language. You can represent chemistry as this form of text known as smile strings. And what you can effectively interpret a chemical reaction as is a translation between 2 languages. If any of you use Google Translate, this is a very, very similar technology. You have an input language, you have a model, and you have an output language. In this case, the input language is the reactants and the reagents. What are the things going into the flask? And the output language is the product. What are the things that come out of the flask? And what we use is an approach known as molecular transformer. And what we're able to show is that we have an accuracy of over 90%, which is currently the state of the art for predicting the outcome of a chemical reaction. It also happens to be the first time that a model has outperformed human chemists who were given the same set of reactions to predict. So this is really powerful. In order to figure out, of the 15,000 submissions that COVID Moonshot received, which one of these should we be selecting to make, and ultimately, therefore, can we find the ones that are most easily makeable so that we can move very, very quickly? So this technology itself of how do you make molecules has now been turned into a product that Posteera offers. So forgive the blatant sales pitch, but I'm really excited just to give you a little demonstration of artificial intelligence in practice. The tool itself is called Manifold, and we actually launched that in the past week or so. So I'm going to try and be ambitious and give you all a live demo of how we have implemented this machine learning tool. So just bear with me while I hopefully bring up another screen. Stan, if you could again just give me a thumbs up if you see the manifold screen here.
Thumbs up.
Fantastic, thank you. Okay, so for those of you who are interested in figuring out how to make molecules, and if you are in the business of making molecules as part of your academic research or as a biotech company, I would highly encourage you to have a look at the manifold tool that we have developed. So what I'm going to do is just kind of run, but let me stick with synthesis because this is the technology that I wanted to talk to you about. So what have we done here? Well, we have basically run our machine learning algorithm in the background to disconnect back to purchasable building blocks, asking the question, can the algorithm find a viable recipe to make this molecule? There are obviously a variety of ways to make a molecule. They're all shown here. And if we go on this explore route, what you'll be able to see is a tree representation of that particular route. And moreover, you can actually look at these individual reactions that are being proposed. You'll be able to get relevant references for that actual synthetic step. So you can actually go in and check the literature to verify whether there is a specific set of conditions, whether there is a specific set of kind of similar reactions that have been done in the literature before. So again, these routes definitely vary by strategic. I guess what we tried to do is offer a strategic diversity in terms of how you make molecules. But ultimately what's happening here is the machine learning algorithm molecular transformer has had a look at these particular reactions and said, I'm going to give it a high level of confidence that this reaction is possible. What you then do is you combine that with a huge engineering effort to search well over 10 billion starting points to figure out how do I take these 10 billion starting points and combine them using these individual reactions. And so the way that we work with clients is they can use this software to then actually be able to export this route. They can then send it to their chemical manufacturer. And what that ultimately does is save a lot of time and a lot of cost for manufacturers and clients when figuring out what is the best way to either find this molecule and purchase it, or ultimately a lot of the time, how do I make this particular molecule? So with that, I'm actually going to end there. Again, just a big thank you to Stan and Suzanne for arranging it. Very, very happy to answer any questions. I believe I've ended just a few minutes early, Stan, so we can hopefully have a bit more time for Q&A. But again, thank you very much.
No, delighted, and thank you very much for the great presentation, and Yang also. Thank you for your presentation. So the way we're going to do it, as we've done in the past, if you would like to enter questions in the chat, we'll get to them. Or if you want to use the little, I think you should have a bar at the bottom, throw up a hand or unmute and ask a question. That can get a little bit unruly, but we can give it a shot. But I'm going to take the prerogative of being the oldest person on the call and ask the first question, if you don't mind. So this is a question for both of our speakers, and we chatted a little bit about it before we got going today. We have a large number of students, postdocs, in this workshop, and I guess my question to both of you is, it seems daunting to jump in and do machine learning and start running these algorithms and coding AI type programs. However, it also is highly profitable, both from a way to get new research done and job prospects. So what advice would you give students, and most of the students on the call and postdocs are from the biology field, for them to try to enter into the AI field and learn how to do that? So that's one question. And then as you answer that question, how did you get into this? Did you learn it when you were a babe, or did you enter it kind of in college? So kind of two-part question, and it's open to both of you. So thank you.
Do you want me to jump in first, Dr. Shen? I'm very happy to. I would have two, maybe three pieces of advice when it comes to trying to delve into the world of machine learning, artificial intelligence. My advice is find problems and write code. Textbooks should always be a backup option. I think you can become very, very, very proficient in the area of machine learning without ever reading a textbook. I cannot tell you the value of actually just staring at a Python script, banging your head against a wall when it doesn't work, when it breaks, when nothing seems to be of benefit. You will learn the quickest by finding a really valuable, useful problem and trying to deploy some form of basic machine learning. If you would like a very practical course that you would just like to take to get involved, I encourage you to look at the Fast AI lecture series. And again, you'll find a similar thesis there that as scary as it is, you will learn the fastest if you write code and just try your hand at different problems rather than reading textbooks. I say that as a mathematician who admires and loves the underpinnings and mathematical frameworks behind deep learning, but it actually hasn't helped me a huge deal, I'll be very honest with you. And so that's my first piece of advice. Get your hands dirty, find a problem. I think Fast AI is a reasonable course to start with. The second thing is that I would encourage you, where possible, to stick to a domain that you understand. I can totally see the appeal of trying to classify dogs versus cats or trying to do next sentence prediction, very familiar machine learning tasks. But I think you should take your expertise in a domain, whether it's biology, whether it's chemistry, because two things. Firstly, you'll be able to be creative in terms of the input to the model. Models are generally only as good as the data you feed them. So if you can feed them something that has a level of expert domain knowledge, that will be valuable. Secondly, when you're evaluating the output of a model, it's also helpful to have domain knowledge to be able to know, does this make sense? Does this not make sense? I can appreciate that most people can see the difference between a cat and a dog, but ultimately, if you're using this in the field of biology or chemistry, it really helps to use your own knowledge as well to be able to assess whether this machine learning model is improving and is getting better. So I hope that helps in terms of just my background. So I began doing machine learning while in finance. I spent kind of 4 or 5 years doing both the kind of learning the mathematical underpinnings, turns out to be not that helpful, but then actually practically doing machine learning and data science for about 4 or 5 years.
Thank you, Aaron. That's really great. And I wanted to hear your advice first. And I just want to echo a few points that Aaron made. First of all, I think our students and the postdocs should be confident that there's the barrier is lower than you perceive. Because I'm not going to argue whether it's easier to teach a computer science student to do pipetting or it's easier to teach biology student to do deep learning. But speaking of deep learning, as Aaron said, there are so many tutorials online now. Sometimes we even use those tutorials to teach ourselves for lifelong learning. So actually, you don't have a disadvantage at all. So the barrier is not as high as you perceive. So I have seen so many students and the postdocs, being able to experimentally generate their own data and then construct machine learning models, generate a new hypothesis, and then do new round of experiments. You actually have advantages. So that's the second point that Aaron was made, was that there are tons of tutorials online, right? So it's kind of hard to, for instance, work on an overly generic problem that your expertise or training wouldn't translate to better outcomes. So instead, there are a lot of Kaggle competitions or Dream Challenge. Many topics are probably directly related to what you do, and you can, actually contribute it to those challenges or the solutions to those challenges in so many ways, you would be more motivated to bypass those, already lower than you perceived barriers, and then you would actually feel the sweetness coming from it. So I think it's completely it's a great process. I think just it's not difficult at all. I believe a lot of students and postdocs have already done it. Speaking of my personal experience, I actually came from the other side. I was a control engineer, electrical engineer by undergrad training. I was doing hardware embedded systems design, but I had a naive love for biology because I knew that I wanted to do mathematics and physics, and I wanted to do control. But naively, I wanted to do control for health systems. So in other words, I wanted to engineer help delivering those perturbations, or eventually called the drugs, to this system. My understanding was very naive, but my passion was never changed. So I actually had to bypass or overcome a lot of barriers from understanding biology. But as I said, this is a lifelong learning process. Even for machine learning, even for deep learning, every year there are new technology comes out. Even computer scientists have to keep learning, and there are a lot of tutorials and there are a lot of materials online. So you don't have a disadvantage at all. You actually might have an advantage sometimes.
Well, thank you both. I think we'll see a lot of new programmers coming out after today, or I hope so. Questions from the audience?
Hi, Stan. Can I ask a question?
Of course.
Hey, great talk. Thank you. So I'm Shuxing Zhang from MD Anderson. So my lab also doing computational modeling and also doing some AI programming to protein drug interactions, protein interactions. So but that's one side. But I'm very interested in the crowdsourcing for the COVID inhibitor development for ARIN. So I think the idea of crowdsourcing was very interesting initially. Like, starting 4 to 5 years ago, these industries like Eli Lilly, GSK, they open these submissions, say, okay, do you have any idea for this specific question? But, after these several years, I don't know how many of them have been successful. But, I always think crowdsourcing with AI will be an interesting combination to go forward. So now you have something to do. You got something like, so I just want to confirm. So you already got a lead compound in March to go forward for IND?
So that is the aim. Like, the program right now is very much in the kind of like lead optimization stages. We have kind of 3 or 4 lead series. Again, all of this information is on our website. You can actually see these results. We hosted a webinar at the end of the year giving our latest updates. But the aim that we are working toward, that we believe we can have ready, is to begin pre-IND and IND enabling studies by the end of March with a compound, the kind of clinical candidates that we will be nominating.
Okay, so I think that's where my so there are several targeting points for the COVID, right? Like the binding process, the spike protein, the entry point interaction with ACE to receptors, so all of these. So there are already a lot of drugs targeting these different points, although they were not specifically designed for COVID-19 virus, COVID-2 virus. But they have been done clinical trials and everything. None of them were very successful. So because I was a coordinator of this principal therapeutic course, so we have a lot of clinicians that come to talk about this aspect. So one of the questions is that although we have a lot of infections, but actually in patients, the death rate actually is not that high many of them actually they got infections, they got okay, and so this give a challenge for clinical trials. So what I'm trying to say is that for lead optimization, for creation, for chemical reactions, it seems is not really a difficult thing to do to get a potent inhibitor in nanomolar, but it's the later round of the like clinical trial design and the after IND and those sort of process. So what do you think with the AI we can do at that stage?
Yeah, that's a great question. So I think you touched on 2 points there. The first was the kind of repurposing effort. I think our conviction is that it's important to have a kind of bioavailable small molecule inhibitor that is specifically designed against the main protease. We think this is important because actually the main protease is relatively conserved across various coronavirus strains. And given that there will most likely be, sadly, future pandemics and future outbreaks of coronavirus, if we can have inhibitors that are specifically targeted for the main protease rather than trying to repurpose existing ones, this will give us a good chance at being effectively prepared for future kind of outbreaks. I'd also say it seems, at least to my mind, right now, a lot of the current antivirals are still administered intravenously. Many of them are still very, very peptide-like. So what we really want is a kind of tablet form, orally bioavailable small molecule. So that's why we decided to work from scratch, from the ground up. Your second point about clinical trials, I think the role for artificial intelligence in clinical trials is more challenging. And I think it's more challenging partially because the regulation in that field, in that part of the drug discovery process, is so much higher, so much more challenging. So to date, it seems the uses of artificial intelligence in the clinical trials, rather than trying to predict clinical outcomes, has been much more, how do we use artificial intelligence to streamline the clinical recruitment process? How can we use AI to effectively improve the feedback that we are getting from the actual trial candidates? I think that it is something that several companies have tried to predict kind of clinical outcomes. I think our thesis in posterior is that biology is just incredibly complex, and it's not something that AI is yet at a point where it can predict the outcome of a clinical trial. So that's why we focus squarely on the preclinical medicinal chemistry effort. Would I love to see that in the future? Absolutely. I don't think we're there just yet.
Thank you. I'm going to open it up to Suman, who's joining us from UT El Paso. Welcome, Suman. I see you have your hand up. I assume you have a question.
Yes. Yeah. Thanks, Dr. Shen and Maurice, for the great talks. And both of you touched on interpretability of the models and also on the confidence estimation, because those are really, really important, not just having the black box models. So I really like that part. My question for Dr. Shen, so you mentioned that you guys are working on the integrating network-based models into generative models to generate drug combinations. So like generating one molecule that works and like getting into clinical trials is very challenging. And so when we have like two drugs, two candidates going into clinical trials, that could be really, really challenging. So how do you see that one going forward? Like what will be the challenges that we would face, and how can we overcome with that?
Yeah, thank you very much, Suma, for the great question. First of all, we were interested in designing drug combinations because of drug resistance. That interest started in 2013 or earlier, 2012, when I started collaboration with clinician scientists as well as biochemists on drug-resistant breast cancer or prostate cancer. So, I think in those cases, drug combination is proved to be a more promising therapeutic strategy than finding a magic silver bullet. So we have to sort of prioritize drug combinations in facing the drug resistance. So that's the motivation. Speaking of the practical challenges that you are bringing up, absolutely, those are paramounting challenges. But there are a few ways we can address that or bypass that, some already mentioned by Aaron. For instance, you can start repurposing. If you can repurpose one drug, then you can repurpose way more drug combinations in more novel ways. So, so that is one way to lower the barriers that you mentioned.
Thank you. Excellent.
Thank you.
Other questions? Either raise a hand, unmute, put it in the chat. If not, I have a whole bunch, but I'll start with one. This is to both of you. It's something that's plagued us in the past. So you're talking about the data sets that you're using to train your models or to develop a model. How in the AI field, and this is outside my expertise, so that's why I throw it out there, is how do you go about cleaning up these data sets? Because in many times, really outlying data points or wrong data points can really screw up a predictive model. Is this an issue in your field? And if not, great. If it is, how then do you clean up the data sets so that you don't really have a wrong model being generated, since you're kind of searching the literature, searching patent databases, and some of that information, will be wrong. So the question to both of you.
Sure, it's a really, really important question. The data in the kind of drug discovery space was not compiled and it was not designed to be used by machine learning models historically. So inevitably, I think for many people, data was kind of just a byproduct of doing experiments. I get some result that I want, and then I just kind of leave the data there. So the first thing to acknowledge is that the data, at least in the area of preclinical drug discovery where we work, can often be very challenging to use. It's both noisy and it's also often in the low data limit. Assay reproducibility is a kind of well-known challenge, and often having a lack of negative data can also be a challenge. In the field of reaction prediction, which Posterior spends a lot of time, people rarely ever report what reactions don't work. And for a model, that's a real shame, because models are very happy to learn from things that do work as well as things that don't work. So that's just kind of me laying the line, saying the first thing to acknowledge is that it is challenging. And the consequence of it being challenging means that domain expertise does become incredibly important. And so within Posterior, we have a very rigid data pipeline, and a lot of that is automated, but we have people staring at data as well sometimes. We have people, chemists, who are looking at the input to models and saying, does it make sense to try and predict the IC50 of this particular assay given the input data that we are feeding this model? So I think, firstly, it is important to have that domain expertise. Secondly, though, on the machine learning side, as an organization, the kind of academic underpinnings of Posterior coming out of Dr. Lee's lab did actually for a while specifically focus on this problem of how can you filter out noise from signal. And there are kind of strict, rigorous statistical methods for determining when a signal is statistically significant. And so there are ways to also try and leverage machine learning to set the bar very high for when one should accept an input of data or when one should trust a model prediction.
Yeah, I think this is a great question. And actually, I think part of my answer probably echoes my first question's answer. You cannot be a great data scientist if you don't understand the data. that includes how the data is generated. So in other words, as a students or postdocs with biology training actually has an advantage. I wish sometimes I have students coming from, I do have students from physics background, but I wish sometimes I have students from biology background and so on, so that at least in the data understanding part, we are not making major mistakes. And not only just curating the data, but also extracting useful information from the data. How we should construct a machine learning model to get that information out also relies domain knowledge. We cannot imagine that a model, like I said before, using so-called attention mechanism would magically and automatically learn where to pay attention to. That's impossible. So that being said, because we are mostly, our group is mostly engineering students, so we focus on very carefully those few problems that we understand well to some extent and already curated to some extent. For instance, the binding DB is already curated to some extent. Of course, after that, we still have to do more curation. For instance, there might be multiple measurements, right? There might be some measurements that's above or below the sensitivity of the assays, right? Those things, at least we have some knowledge to understand and curate. But I think the capability of discovering new biology and designing experiments accordingly definitely starts with the correct understanding and curation of the data. So that needs cross-disciplinary efforts. That needs many talents.
Okay, thank you. We have a question from Emilio.
Yes, thank you for the very nice talk. And regarding what you were saying, I'm in the GPCR field, and I'm really interested in knowing the efficacy of the compounds that we use. Of course, there are some already out there in the market, and some other are being developed. But there is always, when you read most of the papers, there is always a problem when it comes to development. And it's like we recently have discovered in this field, in the GPCR field, that there are two pathways that you can activate, and therefore two measures of efficacy for a compound. So the question is that with AI, is it possible to really differentiate because of the measurements, I guess, you give to the machine to differentiate if it's gonna go into one type of efficacy or to the other type of efficacy? Because maybe, if I understood correctly, I'm pretty new into the AI field. There is a point where you can actually detect that compound could be actually helpful in certain diseases, but then you're also not looking at maybe the other type of efficacy that the same compound can have in that similar system or biological system. So can you both please comment on that, on how can we overcome that type of problem?
So maybe Emilio, I'll have an attempt at just trying to address kind of your concerns there. It seems to me that the assays that you set up to validate efficacy are the real key value there. The machine learning model has a loss function. That loss function basically tells it how well it's doing, and you calibrate that loss function. The model is not going to tell you, hey, have you thought about this other type of loss function? Have you thought about optimizing for a different type of efficacy? So again, this is not something that a machine learning model is going to tell you. So ultimately, whether you fully understand a biological system, to me, is a function of what assays are you running? Are they reliable to test the hypothesis? And then is your machine learning model calibrated correctly enough to optimize for that specific endpoint that you are trying to predict? to me, I guess the very simplistic answer, Emilio, is that you as a biologist, as a practicing scientist, are probably responsible for feeding the model the correct data for it to make the correct predictions. And so it seems to me that setting up the assays at the start as the input data to your model is really critical when asking a machine to predict something.
Yeah, as Aaron said, first of all, we should have some data that encodes information potentially toward what you want to answer. And secondly, you need to construct a machine learning model specifically targeting the answer you're seeking. So both need to be there.
Okay, thank you.
Thank you, Emilio. Ayesha, you have a question? Hi.
Yes, hello. So the problem area that I'm working with, it's a nuclear receptor. And I'm working with, the isoforms. And my goal is to develop an inhibitor. So I am a little bit familiar with, like, the area, the modality I was looking at was more of that of a PROTAC, basically create a molecule that will bring the ubiquitin machinery to basically destroy the protein. So the area that I'm interested in is trying to figure out how do I find that compound that binds to my isoform with very high affinity. And so what you have discussed, how AI could help, my question was around what data would I feed for it to figure out. I mean, I can specify the unique sequences that are unique to that isoform, but then what data do I need to feed? And when you feed that data, do you take into account the structure of the molecule in figuring out the best type binding?
Sure. If I've understood your question correctly, Ayesha, so you're looking to develop a kind of potent selective inhibitor against this is a human protein, a human enzyme?
Yes, it's actually the estrogen receptor beta.
Okay. I'm not intimately familiar with that target, but there are two different approaches traditionally considered. One is a ligand-based approach, and the other is a structure-based approach. Machine learning models have been trained to use both. Your problem is something that Postera deals with all the time. It's what we help our clients solve. We typically, as a company, focus on ligand-based approaches because we ultimately trust the experiments and we trust the data. And although mechanisms of action are important, they're actually not necessary to bring even a drug to market. So we focus on the actual experimental data of compounds tested against that target. So my first question is, has there been in the literature compounds tested against that target in an assay that you deem validated? If the answer is no, this is a novel target that nobody has ever considered, ever seen, or ever tested against, then you're in the second camp, which is, can we use some type of structural information? And the question becomes, do you have such structural information available for that protein of interest? There are methods employed that can try and leverage that structural information to help you design compounds that should be effective against that target. There are some machine learning based ones. There are a lot of non-machine learning based ones. Postera is typically skeptical of some of the non-machine learning based ones based on our experience, but that isn't also starting point. The third avenue is if you have no prior data and you have no structural information, then the solution is just to find a good, diverse screening library and throw everything at that target in an assay. This is dirty, it's not very scientifically impressive, but it's a very good way to get a starting point. And again, this is something that Posterior does a lot. We're big fans of DNA encoded libraries, for example, which have become really advanced in the last few years, which can begin to help you find hits very, very quickly, even if you've got no prior data and no structural information. So that was a very long-winded answer, but I hope it helps in some way.
No, it does. Thank you.
Speaking of the data, there's data that you want to feed to the model, and there as well as the data you want to train the model. So speaking of using the model directly, what data do you need? You definitely need the identity of the protein, which is the sequence. You need the identity of the compound, which could be the SMILES. But for the protein side, you may also want the structures based on what the model is. In our case, we avoided using structures just for the worst case scenario that you only have protein sequence. Now we are talking about models, right? Now, beyond using the model, you also need to construct a model that are able to answer the questions you're asking. And there's a spectrum of methods, models that demand different kind of data that ranges from purely data-driven to purely physics-driven. So, if I go all the way to the physics-driven, I have the protein structure. I run, for instance, virtual screening of a compound, and then a compound library, and then you have a smaller subset of compounds for which you can run the high throughput screening. Hopefully, you have a high throughput experimental protocol, and then going all the way to the data-driven side, that extreme side, that there's no protein structure. There's only protein sequence. And for this protein, we never have seen any compounds with this measurement at all. Then we have to ask, for instance, is this binding affinity only? Then maybe we have seen the binding affinity for other proteins and other compounds. That's what I showed. That's called meta model. So that those are not specifically tailored to your protein, but if there's no data, you can start with that. But if you have even just one ligand or two ligand specific for that protein, you have some measurement, then you can transfer the meta model to your specific target and still get some improvement.
Okay, thank you. Thank you very much.
And thank you both. And because of the hour, Ayesha, feel free to reach out to our speakers or to Suman, who's on the call, or several other people in the area who are kind of expert in this, and see where this may lead. But again, I want to thank Aaron, Yang, both of you for taking the time to present to the group and educate us. And as we slowly move ourselves deeper and deeper into this space, I thank you both, and we'll have you back. So please stay safe, okay, until those COVID drugs are out there.
Thank you, Stan. Thank you, Susan. Thank you all. I learned a lot. I look forward to learning more.
Thanks so much. Bye-bye.
Bye.